Greetings! Thank you for joining us. I'm Kristen McPike, Program and Alliance Manager at the Antibody Society. Today's webinar is one in a series designed to inform and educate our members and the broader scientific community about topics related to antibody discovery and development. Our expert speaker, Dr. Francisco Ylera, will discuss rapid generation of bispecific antibodies through engineered reversibly inhibitable SpyCatcher with us. Please note that the webinar is being recorded. Please add any and all questions to the Q&A box in the viewer, and the questions will be answered at the end of the webinar. Without further ado, I'll turn the show over to our speaker, Dr. Ylera.
Thank you, Kristen. Welcome to the seminar on the SpyLock technology and how it can be used to rapidly generate bispecific antibodies in high throughput. I will start with a few background slides to better explain the setting and why we developed the SpyLock technology. At the Bio-Rad site in Munich, in Germany, we generate custom monoclonal antibodies by phage display for research and diagnostics for our customers for about 21 years. And 2 years ago, we added the therapeutic discovery, antibody discovery as a service with the Pioneer platform. The newest addition to this platform is SpyLock. It's based on the SpyTag technology, which is a very powerful and versatile technology. It consists of the SpyTag, a small peptide tag, and a SpyCatcher, a small protein. And if you mix these two, they spontaneously, autocatalytically form an isopeptide bond between lysine side chain in the SpyCatcher and an aspartic acid side chain in the SpyTag, shown on the right. This reaction is very fast and quantitative. Within minutes, you get your ligated proteins, and usually within 10 minutes to an hour, the reaction is complete. The reaction is specific for these two partners, so you can have this reaction within cells or in lysate, and it happens only between the SpyTag and SpyCatcher. About 5 years ago, we launched our Trailblazer technology, the modular antibody assembly platform. We added the SpyTag to our antibodies here to the Fab, to the C-terminus of the Fab heavy chain, and we generated a set of different SpyCatcher modules, the SpyCatcher, the dimer, the bycatcher, and we fused the SpyCatcher to the Fc domain. And here we have the human IgG1, 2, 3, 4, IgA, IgM, mouse version, a rabbit version. They are all in place and also available through our catalog. And in addition, we introduced cysteines into the SpyCatcher for site-specific labeling. So we also have a set of labeled SpyCatchers in the catalog. And with these catchers, you can very easily and rapidly generate all kinds of different antibody formats. So you purify your Fab once, and then you mix it with a catcher, which is of interest, and you let it sit for an hour, and you have your product, and you are ready to go for your assay. And here it's important to emphasize that it's a site-specific reaction at the C-terminus of the heavy chain. So if you look at the conjugated antibody, then each Fab carries a SpyCatcher after the reaction, and the SpyCatcher, each SpyCatcher carries a label, and there's no modification of the binding site, so all antibodies are still fully active. So as a consequence, performance in assays is usually much better than with conventional labeling chemistry. And of course, it's also much simpler and faster, and this is especially true for the synthetic IgGs. You can take the whole output after selection and convert it within 1 hour into this synthetic IgG format instead of recloning, transfecting, expressing, purification, which all takes weeks. So it is much faster and also more economical because you can do it in small scale or in bulk as needed. So this platform was actually launched already 5 years ago, and it's since then a big success for our custom antibody customers. But we also use the SpyTag for the selection of antibodies with a technology which we called SpyDisplay. Here we have a phage MIT which carries only the Fab and no other elements for phage display. So we can actually clone the library, the antibody library, into the expression vector. We use an E. coli with a modified genome, which carries a gene for the SpyCatcher P3 fusion. And then the bacteria produces new phages, which carry a SpyCatcher on the surface. And from the phage MIT, we get our Fab with a SpyTag. And then these two ligate within the cell, and we get a covalently displayed Fab on the surface of the phage. And the advantage of SpyDisplay is that it is, first of all, fast. We can do 1 panning round per day. And between selection and screening or production, we do not need any subcloning steps because the library is already in the expression vector. And it's also a very efficient method for the selection of high affinity antibodies because it's a real monovalent display. And in combination with the Pioneer library, we get very good affinities out of our selection campaigns. And SpyDisplay is used for the Pioneer library. It's a new library in which we inserted all our experience. It's a curated antibody phage display library. And with more than 200 billion unique antibody, it's one of the largest functional antibody phage display libraries. That's shown here on the right, the green bar, compared to the competitor libraries. It is probably about factor 2 larger than the next largest library. But it's not only large, it's also optimized for phage display so that we have sequences which perform well during the selection procedure. But of course, since it's meant for therapeutic antibodies, it also was optimized for a good developability of these antibodies. Together with SpyDisplay, we can select antibodies very quickly and get high affinity antibodies out of it. And with the Trailblazer platform, the modular antibody assembly platform, we can screen and characterize these antibodies very rapidly. And as a result, we get a very high hit rate and a very high diversity out of our antibody selections. We have excellent affinities with more than 20% of the antibodies having sub-nanomolar affinities directly out of the library. We showed previously that we can generate functional antibodies against difficult targets like GPCRs, including selections on cells. And the resulting antibodies have very good developability parameters. So previously we showed that the resulting antibodies compare very favorably to antibodies in the late stage of the clinical trials or even on the market without any further optimization directly out of the library. And recently this year, we added the rapid access to bispecific antibodies for the identification of the best antibodies pairs with the SpyLock technology. So the SpyCatcher reacts with the SpyTag and forms the isopeptide bond. So we took the SpyCatcher and we mutated single amino acids to cysteine. We did a cysteine scan in the ordered region. So we generated about 80 mutants carrying one cysteine each and tested these for their capability to react with the SpyTag. And basically, they all do react with the SpyTag, of course. Shown here for set of mutants. So we have the SpyCatcher band at the bottom. Then we added MVP with a SpyTag, the band at about 40 kilodalton. And on top, we have the ligation product. And you see for the K31C, there is a band missing. that is the lysine, which forms an isopeptide bond. Of course, if we mutate this one, then it does not react with the SpyTag anymore. The same is true for the glutamate, which is required to catalyze the ligation reaction. So now we have a SpyCatcher with a cysteine in there, which has a free thiol group, and we can modify it, form a disulfide bond with a small disulfide bond forming reagent like Ellmann's reagent. That's shown here in the middle. And the product is shown on the right. It's a SpyCatcher, which carries this TNB group at the cysteine. And then we retested for the ligation capabilities with the SpyTag. And we found that there are some mutants or modified mutants which do not react anymore. That's shown on the gel at the bottom left. Again, at the top, it's a ligation product, the band. And for the S59C, you see that the band is missing. So that is a mutant which does not react anymore. So we can actually modify this mutant and make it inactive. We actually found 8 of these mutants, which we call SpyLock. So we can convert them from a reactive SpyCatcher to an inactive SpyCatcher. The reactive one we call open SpyLock. The inactive one we call closed SpyLock. And as you all know, a disulfide bond can be easily cleaved with a reducing agent. So we can convert the closed SpyLock quickly into an open SpyLock, which then is reactive again. And that's shown here on the gel at the bottom left for the 8 SpyLock mutants. The first lane of each triplet is SpyLock alone. The second one with addition of MVP SpyTag. And you can see that there is no ligation product. And when we add TSAP as a reducing agent, then in the third lane, we get the ligation product on top, the topmost band. So next, we actually checked whether it's an effect of this charge of the TNB group, which we added. So instead of TNB, we used an activated biotin, HPDP biotin, which we attached via disulfide bond to the catcher and tested it again in comparison to the TNB locked SpyLocks shown on the gels at the bottom, always comparison TNB versus biotin. And basically there is no big difference. There's some minor difference for some mutants, but in general, one can say that it's not the charge which is important. It's probably the size which is added to the cysteine. So here's a kinetic gel for the reaction. The first block shows the reaction with the SpyTag for the locked closed SpyLock, and you can see there is no ligation product. Only after 16 hours we get a faint band, so that basically shows that the SpyLock is not inactive, it's just strongly inhibited. And 16 hours is far longer than necessary to wait. And if we add reducing agent next to it on the right, then you can see that basically within 1 hour the reaction is complete, 1 hour or a bit more. So the reaction speed of the SpyLock is actually a bit slower than for wild-type SpyCatcher 3, but the speed is still sufficient that we can get quantitative coupling within hours. So no extended incubation times required. The SpyCatcher is actually consists of beta sheets which form a beta barrel, but not a complete beta barrel because the SpyTag is missing. And because it's not complete, it actually has a reduced thermal stability, which you can see in the diagram on the right, the red curve measured by nano-DSF. So you can basically see that the onset of denaturation starts below 30 degrees already, with a peak somewhere in the mid 40s. If we now add the SpyTag to the SpyCatcher, then the beta barrel is closed, which increases the stability of the protein dramatically. That's shown with this black curve. Here we have a onset of the denaturation at 85 degrees or so. So a dramatic shift. If we now look at the SpyLogs, then these are the gray bars in the bar diagram. We have the wild-type SpyCatcher on the left and the 8 SpyLog mutants in gray next to it. So they all but 2 have a lower melting temperature, which basically indicates that this cysteine mutation destabilizes the structure of the SpyCatcher. The S59C mutant is actually just one mutation, one atom different. The oxygen in the serine mutated to a sulfur in the cysteine, so quite a minor change. And here you can see that the resulting melting temperature is actually slightly higher, but not a big difference. If we now modify the SpyLogs with a biotin, it further destabilizes the structure, which we see in a decrease in the melting temperature, which is shown with these red bars. For some, it is a very dramatic decrease of melting temperatures. For others, it's not so dramatic, but the interesting thing is that we still seem to have a defined TM temperature for these closed SpyLogs. So the SpyLog still seems to be folded. And if we add a reducing agent to it and cleave off the biotin, then we get the blue bars, the blue for the melting temperature, which are similarly to the open SpyLogs before modification. So after treating with a reducing agent, the SpyLog seems to go back in its original conformation, has the same melting temperature, and as I showed before, is active for the SpyTag ligation. We actually looked at the structure of SpyCatcher and where these mutants, this mutated amino acids sit. And first we saw that 6 out of these 8 amino acids are hydrophobic amino acids. And 5 are actually pointing towards the inner hydrophobic core of the beta barrel. So next we were wondering if the resulting SpyLog after SpyTag ligation carries a free cysteine. How does it actually affect the product? Does it dimerize over time via oxidation, or what happens? So we looked at the SpyLog in comparison to a SysCatcher, which carries a cysteine at the end terminus, so away from the reactive site, easily accessible. So first we thought fresh SpyLog and SysCatcher, and on the gel you see that it runs as a monomer as expected. If we let it sit for 24 hours and load it on the gel, then we see dimers for both SpyLog and the SysCatcher. If we try to let this dimers react with the SpyTag, the MVP SpyTag again, then we see that the reaction takes place for the SpyCatcher with the N-terminal cysteine, as we would expect. That's the band at 130 kilodalton. But we do not see this band for the dimerized SpyLock, which is not surprisingly, because I showed before, if we add this biotin or TNB to the cysteine via side-by-side bond, then we block the reactivity. So if we add a SpyCatcher instead, it's even bigger, then it's clear that we also block the ligation reactivity. But the interesting part is the last two rows. So here we coupled the monomeric SpyLock or SpyCatcher with the N-terminal cysteine to MBP, and then let it sit for 24 hours. So for the cis-catcher, we see what we would expect. It couples with MBP and then dimerizes. We get the band at 130 kilodalton, but the SpyLock only couples with the MBP SpyCatcher, but doesn't dimerize anymore. And if you look at the structure again, then it's actually not so surprising, because I mentioned that the cysteine is directed towards the hydrophobic core, so it's inside the beta barrel. With the addition of the SpyTag, we close the beta barrel, and the sial group is actually not really accessible anymore. So dimerization is not really an issue for the ligation product. So what can we do with the SpyLock technology? We wanted to use it for bispecific antibody generation. And for that, we generated a fusion protein of a wild-type SpyCatcher and a SpyLock. The SpyLock is inhibited with biotin. And if we now add a protein, an antibody with a SpyTag, it can react only with the wild-type SpyCatcher because the SpyLock is closed. But we can later unlock it and use it for a second coupling step. And that's what we do. So we start with our SpyLock catcher. We add the first Fab with the SpyTag, the green one here. It reacts to the intermediate product. And then we add a reducing agent. We remove the lock, add a second antibody shown here in blue, and we get our bispecific antibody. It's a very simple and fast protocol. It takes about 90 minutes. It can be done in high throughput, and we usually get purities of about 90%. It basically depends on the accuracy of the concentration determination of the antibodies, because you want to mix the SpyLock catcher and the antibody in a 1-to-1 ratio, so that you really get only the bispecific antibody. If 90% is not sufficient, we also have a high purity workflow. Here we add the first antibody in excess, so that all SpyCatcher sites are definitely coupled to the Fab. And then we purify the intermediate product on streptavidin beads. The excess of Fab is washed away, and we have only our intermediate product. And then we elute it with a reducing agent, TSAP, and at the same time we unlock the SpyLock, and we add the second antibody, the blue one, in excess. It reacts completely with the SpyLock and add it to SpyCatcher beads. And then the excess of Fab with the SpyTag reacts with the SpyCatcher beads, and our bispecific antibody stays in the supernatant, and we get purities of about 99%. Also, this protocol is quite fast and is suitable for high throughput, just that the yield is slightly lower because of these purification steps, and it takes a bit longer. Here on the gel, I want to show the purity of the resulting products. So if I would do it for a bispecific antibody, it would be difficult to show whether it is a homodimer or heterodimer, and therefore I used Fab with a SpyTag for the first coupling step and MBP with a SpyTag for the second step. On the right, you can see with an asterisk that's a homodimer for the Fab, monospecific bivalent Fab, and with two asterisks, it's a MBP dimer. That are basically the two byproducts which we could expect. And if you look for the rapid protocol, we basically see the Fab homodimer, small band. Above it is our product, which we want to have. And for the two asterisks, there's a very faint band for the MBP homodimer. So these are basically the bands marked with the asterisk are the products which we don't want to have. Ideally, so if these contaminations are not acceptable, then we can use a high purity protocol shown with the red rectangle, and there we only see the ligation product for the heavy chain and the light chain, so no contaminations by these two byproducts anymore. One question which I was asked in the past was, I mentioned the reducing agent several times. Is it actually compatible with antibodies? Because antibodies contain also disulfide bonds. The interchain disulfide bond between the light and heavy chain in a Fab is actually not really required. It's also not present in UCAL and Pioneer antibodies. The light chain and the heavy chain, they also stick together if this disulfide bond is not present or if it is cleaved. So there we do not see any issues. So what about the intrachain disulfide bonds? Do they get cleaved? To find out whether that is an issue, we tested Fabs. We looked at the affinities after incubating the Fabs with 5 millimolar TSAP for 16 hours. That's basically much longer than it's needed for this workflow, just to be on the safe side, and measured the affinities after this treatment or in PBS. And that's what you see here on the right, the red bars in PBS and the blue bars with the reducing agent, and there is basically no difference. We even extended the incubation time and stored Fabs for 1 week in 5 millimolar TSAP and compared the performance in Ylera. That's shown in the diagram at the bottom. The red and magenta curves are basically on top of each other. The black and gray, the same. The blue and the light blue belong together. There we see a slight difference, the same for the green and the light green curve. So after 1 week, we see for some antibodies a slight decrease in activity, but only a slight decrease. And basically a week is far longer than what is needed to generate these bispecifics. As I showed you, it's possible to do it in 90 minutes. But furthermore, we actually quench the TSAP after the reaction is finished by the addition of PEG azide, which reduces the free TSAP by about 97%. So there's basically hardly any active TSAP left, so the stability is not really an issue. Next, we actually looked at the bispecifics, whether they perform as we would expect it, and we started with an Ylera. Since we have a lot of anti-idiotypic antibodies, we started with 2 anti-idiotypic antibodies against ocrelizumab and dupilumab. So we coated, in this Ylera, we coated ocrelizumab, added the bispecific antibody, either with a rapid protocol in black or the pure protocol in red, and detects with dupilumab, which is conjugated to HRP. And you can see we get these two nice curves as expected. If we add a bivalent monospecific dupilumab antibody, so both arms binding to dupilumab only, then it's a green curve which does not go up. So which basically indicates that we do not really have monospecific antibodies in our mixtures, which are messing up this assay. Even with a rapid protocol, we get a nice curve. Yeah, we get the results we expect. We also did the control the other way around. We coated ocrelizumab and detected with ocrelizumab HRP. So here the bispecific antibody should not give a signal. So the blue and light blue curves stay down, and only the homodimeric anti-ocrelizumab antibody gives a signal in magenta. So far everything good. Next, we wanted to confirm that this format is actually also suitable for testing in a reporter cell assay. For this, we chose PD-1 and PD-L1 as targets. PD-1 is an immune checkpoint, which is a common target for cancer immunotherapy. Basically, PD-L1 is a ligand to PD-1, and if they interact, then it leads to an inhibition of the T-cell response. So the PD-L1 on the tumor cells binds to the PD-1 on T cells, and this way the tumor evades the immune response. So it's known that there are quite some antibodies under development and in the clinic, as well as on the market against PD-1 or PD-L1. And it's also known that bispecific antibodies against PD-1 and PD-L1 show an enhanced anti-tumor activity in mouse models. And that's why we chose this model. We selected 4 PD-1 antibodies, pioneer PD-1 antibodies, and 9 PD-L1 antibodies, and generated bispecifics, so 36 different combinations, and tested these in a reporter cell assay. Basically, if the interaction of PD-1 and PD-L1 is inhibited, we get a luciferase signal. So we did that for our 36 candidates, that's shown in the diagram on the left. But more interesting or better to see it in the diagram on the right. In blue is the PD-1 antibody alone. In red is the PD-L1 antibody alone. In green, we have the mixture of these two, so not bispecific, just the mixture of these two antibodies. And in black is our SpyLock bispecific antibody. So you can clearly see the enhanced potency, and yeah, it shows clearly that the SpyLock bispecific antibodies can be used in a reporter cell assay like this one to find out which are actually the best performing candidates. And that brings me to the conclusion. SpyLock introduces a simple control of the ligation activity into the SpyCatcher. We can generate bispecific antibodies with high yield and purity of more than 90%. We can generate a large number of bispecific antibodies in parallel and high throughput for selection of the best pairs, the most interesting pairs in cellular bioassays. And currently we are actually comparing these two conventional bispecific formats. So we didn't manage to get this data in time for this webinar. So watch out in the future for this comparison. And Also important to emphasize is, especially for those who are not so familiar with bispecific antibodies, usually you cannot take the best antibody A and the best antibody B and make your bispecific, but you have to screen a range of different antibodies to find out which have the best affinity or the most suitable affinity or epitope. And if you start with 10 antibodies A and 10 antibodies B, then you have basically 100 combinations which you can make. If you do it in a conventional way, it takes several weeks to get these, whereas with a SpyLock technology, you can generate these hundreds or even more bispecifics in a day, and on the next day you can screen them in your cell assay, and before you have even cloned the bispecific constructs for this other approach, you have the results already, which ones look promising. So, and with this, we actually added the fourth technology to our Pioneer antibody discovery platform. So, in addition to the very large Pioneer library, our very fast and efficient spy display selection technology, the modular antibody assembly platform Trailblazer for the rapid screening and characterization. We now also added the SpyLock technology for the rapid screening of bispecific antibodies. Basically trying to accelerate the antibody discovery from discovery to lead generation, trying to minimize and the time required, and of course also trying to increase the quality of the resulting products. And with this, I say thank you for your attention, and I'm happy to answer your questions.
Thank you so much, Dr. Ylera. So just a few questions. First, can the SpyLock bispecifics format be used for drug development?
So the SpyLock bispecifics cannot be used as a drug by itself, because the SpyCatcher is a bacterial protein. It comes from Streptococcus pyogenes. So if you would use this bispecific antibody in humans, then you can expect an immune response against these. So it is basically just a screening tool to accelerate the developability, the development of your drug candidate. As I said, if you have 100 combinations, instead of waiting for weeks to generate bispecifics in an IgG format, you can do the whole generation and screening in 2 or 3 days, depending on how many assays you plan to do. And then the best candidates have to be converted into the final format.
Thank you so much. So next, can I just buy the SpyLock and test it out myself?
Yeah, so first we launched the SpyLock technology as a service for our UCAL and Pioneer customers. But we would like to see that this technology is actually used widely. So if you are interested, it's currently not commercially available through the catalog or so. So if you are interested and you would like to try it out, then simply contact me and we will find a way to get the BioCatcher to you.
Thank you so much. Is the geometry of the bispecifics the same as a bispecific IgG?
It's obviously a different protein, so the geometry is not the same. We showed in these assays that it works as expected in this Ylera and the cell-based assay, but it is not the identical geometry. So that's why we want to compare it next to an IgG format. And as you probably know, there are also different formats for bispecifics. So we are still working on it to add more data to it. And yeah, we believe that it is a powerful tool to screen for the first selection to narrow down the interesting candidates.
Great, thank you. So when do you expect to have that next round of data available?
It's probably towards the end of Q1. I hope to have it ready. Yeah,
Great.
if everything goes smoothly.
And the last question, do you have a SpyLock that includes the FC domain?
No, that's something we are thinking of, but we cannot offer that one yet. That will also take a bit longer before we have it and have properly characterized it. So our SpyLock bispecifics currently do not contain the FC domain.
Understood. Great. Okay, well, thank you again for this insightful presentation, Dr. Ylera. The on-demand version of this webinar will be available very soon, and a link will be sent by email to everyone who registered. Feel free to watch this or any of our other on-demand webinars when it's convenient for you on our YouTube channel. Thank you again, and have a great rest of your day.